Okay. So the project is under the topic of bioinformatical approach in identification of a possible biomarker in signaling pathways on tumorigenicity of cancer initiating cells. So this is the content. So our aim is to find a biomarker which is common for the cancer types of our interest, which are lung cancer, breast cancer, and colorectal cancer. So the objectives of this study are to analyze and determine significant differences of gene expression between the cancer types we chosen and with their subtypes. The second one is to analyze molecular differences of expression between cancer and the control groups. The last one is to analyze different types of drugs used for the treatment of the three cancer types. So, cancer is the uncontrollable growth of abnormal cells in our body, and a tumor has a special type of cells which are known as tumor-initiating cells or cancer-initiating cells. These cells has a self-renewal and tumor-initiating capacity. Conventional cancer therapy only targets the cancer cells in the tumor, but the cancer-initiating cells remains without any harm, where it can give rise to the cancer again and again. This is the importance of targeting cancer-initiating cells. So, according to World Health Organization, the most common cancers all around the world are lung cancer, breast cancer, and colorectal cancer, respectively. So, this is the reason why we chosen these three types. So, as I mentioned before, uncontrollable growth of cells observed in lungs, breast, colon, and rectum in their respective cancer types. So, targeting the cancer initiating cells through their specific pathways is what we are looking for. So the the specific pathways will be discussed by Vindhya.
Thank you, Avinash. So cancer stem cells are influenced by many highly conserved signal transduction pathways that aid in tissue development. However, for our project, we selected WNT, Notch, and the hedgehog pathways as they aid in self-renewal and lead to tumorigenicity. In the first pathway that we selected, the Notch pathway, it is a self-fate determining pathway that contributes to angiogenesis and tumor immunity, and thus it delays the cell differentiation and prolongs the proliferation. It comprises of 5 Notch ligands and 4 Notch receptors. The interaction of these ligands and the receptors triggers the proteolytic cleavage of the receptor, resulting in the release of intracellular domain to be bound to the transcription factor to express the target genes. In the WNT pathway, the ligands are bound to the frizzled receptor and activates the DBL protein. This leads to the degradation of the disruption complex, accumulating beta-catenin in the cytoplasm, and then it binds with transcription factors to transactivate the targeted genes. In the hedgehog pathway, the ligands are bound to the PTCH receptor and relieves the inhibition of SMO. This then inhibits the cleavage of the GLI transcription factor and activating it, leading to the initiation of the transcription of the target genes of that pathway. Now to explain the inputs and the methodology, I hand over the presentation to Diniti. Over to you.
Thank you, Vidya. So, considering the methodology, we have chosen 3 bioprojects for each cancer type, which are breast, lung, and colorectal, as Avinash has explained before. So, when selecting the samples, we have selected biopsy samples for higher reliability, since biopsies are the tissue samples taken from a living body rather than selecting those commercially available cell lines. The instruments used were Illumina, and for this transcriptomic analysis, we have used paired cDNA samples. Previous one. The slide shows the control and patient samples we have selected. For breast cancer, 2 subtypes were selected, which are TNBC primary tumor subtype and ER positive and HER2 negative primary tumor subtype. And the sample selected from mammoplasty breast tissues is the considered control. For colorectal tumor derived from primary colorectal tumor is the patient sample, while epithelial and stroma derived healthy tissues are the control samples. Same goes for lung cancer, while lung cancer control sample is epithelial tissues and lung tumor tissue samples are the control. Moving on to transcriptomic pipelines we have used, there are two pipelines used, which are RNA-seq pipeline for sailfish algorithm, shown in figure 1.2, and differential expression pipeline for DSEQ2 algorithm, shown in figure 1.3. So, explaining these algorithms, sailfish is used to quantify the previously annotated RNA isoform abundance from RNA-seq data. This is essential for gene expression levels, and expression table can be obtained from this algorithm. Moving on to DSEQ2, it is used for differential expression analysis. Differentially expressed genes are identified for an expression pattern for differentiation between groups. So to explain about the results, I'll call upon on Dave.
Thank you, Dinity. By running both Sailfish and DSIC2 pipelines, we were able to obtain a KEGG pathway in JAK-STAT signaling pathway. So JAK-STAT signaling pathway shows a significant difference in gene expression, differential gene expression, rather in compared to patient and the control. So let me briefly describe the JAK-STAT pathway. So JAK-STAT is not like any other pathways. It is not considered as the key pathway, but it includes ligands, receptors, and intracellular signaling cascade. So there are receptors such as GP130, GP140, INFR1, IFNR2. So the question is, what is JAK? So JAK represents Janus kinase. It's not like any other kinases which utilize ATP to phosphorylate. So it has a auto-phosphorylation cascade. So cascade. So the next protein is STAT, which means signal transducer and activators of transcription. So there are 7 different STAT encoding genes. So some of STATs are also post-transcription modified for the form of additional isoforms. So they are also can be considered as transcriptional factors. So when the receptors activates JAK will be autophosphorylated. So in turn, it will activate several pathways. So such as PI3K, RAS, and STAT pathways. So after STAT phosphorylate and produce homodimers, it can enter to the nucleus to activate gamma activated sequence elements. So it has a function in cell viability and survivability and etc. In this case, what happened is when in upregulation of JAK will leads to a cell cycle progression, and what will happen is the cells will get developed rather than undergoing apoptosis. So in this case, and the other thing is this JAK-STAT signaling pathway shows crosstalks with other signaling pathways which will initiate other metabolic pathways such as protein metabolism, carbohydrate metabolism, and other pathways. So considering this JAK-STAT signaling pathway is very essential when it considered for treatments in cancer. So I'll move on to to improve this, we just ran a sailfish pipeline. To improve this, we have to undergo with PCA quantum norm and a bow tie top hat. After running those pipelines, we can come to a good conclusion with with including other bio projects. We can come to a main conclusion. So I'll hand over to Tharinder to proceed with conclusion and references. Thank you.
No, thank you, Dave. So, more informative biomarkers are essential for the interpretation of the certain type of the biomedical conditions. These biomarkers used to measure the biological states. Nowadays, simple and complex biological states measured through the use of single or multiple biomarkers. Confirmations and the clinical validation of the particular new biomarker is long. It's a long procedure. It take a longer time to validate. Biomarkers can be used to find out what will happen to you when you use the new treatment or new drug, or don't use the treatment. And the risk of the development certain medical condition can be identified with biomarkers. Um, let's move into field application of these studies. Next. Uh, Sorry. Uh, Field studies of these condition uh, have to be more saturated with more data, and the visualization technique have to be increased and more specific. Uh, so these data can be used to develop a new testing kits for cancer subtypes or particular cancer types. Uh, these studies are important for reduce the higher costable diagnostic technique. Diagnostic accuracy increases so that it will decreases the uses a non-effectable treatment method. Let's move into next slide. These are references, uh, and thank you for listening.